Hi, this is part two of Human and Microbes, and it will be the completion of the presentations for this course. So, today or tonight, I'm going to be talking more about stealth pathogens and specifically some examples of types of stealth pathogens. So, anyhow, and the modes on which, the ways in which they work. Okay, one type of stealth pathogen is a pathogen that grows within the host cell so that the immune system cannot see them. And a genus of bacteria that are very good at doing this are your Bartonella. And in this picture here, we have 4 species of Bartonella that are infecting a host cell. But even though they belong to the Bartonella genus, they infect the host cell in different ways. A couple of the examples there, we've got the Bartonella henselae, which is up in the circle, and that infects either singly as a single bacterium that will be taken into the host cell, or a group of them, a bundle of them, may be taken in or infect the host cell. Then we've got the rickettsias down there on the bottom. Again, they will enter one by one, but then once they do enter or infect the host cell, they form a protein-like tail made of actin that will propel it or move it around the cell. We've got the Wolbachia, which is on the bottom left. Once they enter the host cell, they may directly move to the nucleus and affect the gene expression within that cell. And then finally, we have the Brucella up there in the upper left-hand corner. They enter, and again, they enter different ways. They actually may be, in this case, the cell enters on some lipids known as a lipid raft. So you don't have to know all these 4 examples. I would say just focus on the Bartonella henselae up in the right-hand corner. I'm going to talk a little more about that in the next slide or two. All right, so again, Bartonella, here's one of the first slides, and we're talking about these species henselae. Now, the interesting thing about this bacterial cell is that it is carried by a flea or a tick, and it is also the cause of cat scratch disease. So that flea or tick will bite the human or the host, and the bacteria living within the flea will infect one type of cell first. It's called its primary niche, and it is the endothelial cells of the blood vessels. So that bacteria, once it is injected into the host, will infect the first cell, which is your endothelial cells that line the blood vessels. So after a few days there, what happens is that bacteria will be released into the bloodstream. And when they released into the bloodstream, one of 2 things may happen. The first thing is that they may reinfect another endothelial cell, or they may infect the red blood cells that are flowing along within the blood vessel. So the interesting thing about this Bartonella, the takeaway, is that it is a stealth pathogen, but that it actually has two cells in which it infects. And the thing about the red blood cells, if they are infected, they will stay there and pretty much be dormant And it is really a neat way or neat strategy that the bacterial cells have formulated over the years, because if they're within the blood cell, that blood cell then, um, if it is taken up by another flea, uh, will take up that blood cell, and then in that way the bacteria can be given to another host. So there's an example of a bacteria that is a stealth pathogen. And most of the times, these stealth pathogens do not affect are very low, um, they very weakly affect the immune response. But anyhow, you do have other examples that are not bacteria that will be pathogens that will affect the immune response, and those are your frontal pathogens, and you may have some that are stealth. So we're moving out of the prokaryotic uh, category into the eukaryotic. So I just want you to be aware that eukaryotic parasites can also um, affect the host cells. And I think I may have covered this in a previous talk. But these are some of the examples. You have fungi, you've got protozoa, eukaryotic cells that will also can be pathogenic. And we have the worms. Okay, we used to do a lab on two of these, the Babesia and the Dirofilaria, but we didn't do it this semester. That's why you see it in red. Okay? All right, so anyhow, now another type of pathogen is the acellular pathogens, are viruses. And I remember when there was a PowerPoint a few semesters ago that the viruses were summed up as bad news wrapped up with a protein coat. But again, we are finding out more and more that viruses, it isn't just black and white either with the viruses, because we have found that these remnants or little sequences of DNA that belong to viruses are actually a part of our human genome. Again, remember that our human genome, our cells are mostly consist of non-coding DNA, the chromosomes, and also, like I said, some of these viral derived DNA sequences. Now, the sequences, even though they come from viruses, for the most part are non-functioning. They're just bits and pieces of that virus's genome. But it just shows that over time, through evolution, we have acquired some of these viral sequences into our genome. Some other things about viruses here, if you'll see on the right-hand side, we've got those colored bubbles. We do have some that are part of our genomes that do operate as retroviruses. They are remnants of retroviruses. We also have in the green bubbles on the right-hand side there some of the common knowledge we have of viruses, that they can contribute to persistent infections. Also, you may have your acute primary infections caused by viruses. Also, we know that there are new viruses that are always popping up that occur in our history. Now, on the left-hand side, we've got the blue bubbles, and that's just talking about the viruses of bacteria, because this is microbiology. And again, we have bacteriophages that are commensal, so they do not really harm us or harm bacteria so much. We have new bacteriophages that, through mutation, they are being made. But also we have bacteriophages with pathogenic potential or antibiotic resistant genes. So as they go from one bacteria to another, they may be picking up some of these antibiotic resistant genes, which will um, which will affect the bacteria, which, if that bacteria infects us and is pathogenic, can be detrimental to us or can cause us harm. So this is just, there's more to viruses than just good and bad. There also is some unknown. Okay. Some viral infections, many of them cause the primary or acute diseases, but there are some that overactivate our immune response, and they cause something called a cytokine storm. So an example of this is the influenza virus. And how that works is, I'm not going to go into great depth with it, but how that works is you're infected initially by that virus. So some of the responses may be, a respiratory illness, but that is usually cleared out from us, from the host. You may get complications of pneumonia or acute respiratory distress syndrome. What happens though with these viruses is they may also bring about a very acute, like I said, overreactive immune response. That is your cytokine storm. So when the virus infects us initially, you are bringing in your innate and adaptive immune response. Along with that, with the cells, there also is a very high amount of cytokines released, which can switch on a lot of the extreme immune responses, and this can lead to death. Another thing about the viruses is some may actually establish latency and will be reactivated later. So this one, I ask that you know a little bit about the Bartonella and about the human herpes 3 virus. Human herpes 3, that is the family in which the virus belongs to. So we all know this as varicella, which is chickenpox. Your primary infection with this virus is chickenpox, which causes a rash, but in some cases it may lead to complications. It usually infects young children, and if those children are healthy, they will be cured or just get rid of or overcome the chickenpox. However, some of those viruses will become latent, can and will become latent, and they end up becoming latent in cells in the cranial and spinal nerves. So they become dormant in these cells. At some point later on, they may be reactivated. They don't know quite why. They do see that, or they do think that it's because our immune system is not up to par. So you will see this happen many times in older individuals or individuals with a compromised immune system. And when those are reactivated, they will then form a rash, usually along nerve pathways, and again you will see the rash, and that will be as shingles. So anyhow, there are complications with the shingles, and this can occur without a rash, but some of those complications are listed in the right hand box down there. Hepatitis, pancreatitis. You do not need to know the complications of these of this virus. I would just ask that you know the name, varicella zoster, and it's chickenpox, which may later, when it's reactivated, become shingles. And if anyone's had that, or what I hear is that it is very painful as shingles. Another virus, human herpes virus 4, is also known as the Epstein-Barr virus. And initially, when it establishes infection, it will establish its infection um, in the B cells, so type of lymph cells. And if it runs its course, it is an acute infection initially, and we know it as mononucleosis. But at some point, these viruses will become latent in the memory B cells or the lymph cells, and they will rest in those cells in the lymph nodes, peripheral blood, and bone marrow. Now, what happens to these latent infected memory cells will be explained a little in this slide here. So what they are saying here is that epigenetics influences the Epstein-Barr virus disease. So epigenetics is just a big word for the study of changes in organisms caused by modifications of gene expression. So what they have found is this Epstein-Barr virus in the latent stage within the B cells may cause a modification or may affect the expression of certain genes within that B cell, that human host memory B cells. And they have linked this to multiple sclerosis, to systemic lupus, and rheumatoid arthritis. They have linked it, this latent EBV virus, they have linked it to autoimmune diseases. Okay, this slide here is again talking a little more about the Epstein-Barr virus and its cousin CMV. They're both very closely related. And what they say more times than not, these viruses do infect lymphs, but they can also infect other cells. And most of the time, it's in equilibrium. They don't know at some point what triggers the epigenetic response that will trigger an abnormal function in the host cells. So at what point do these viruses which have infected the host cells, at what point do they cause the change in the expression of genes of those host cells? So this is some research that is still needs some study. Now they also have studied the Epstein-Barr virus in co-infection. So what a co-infection is that you've got the virus with another pathogen or another organism, bacteria, within an environment. So what they did here in this study is that, first of all, there was a study done in which they had a suspension of lymphocytes and the Epstein-Barr virus from individuals, healthy individuals. This blood was suspended into a broth. And they would check the viability of it. And normally, your white blood cells or the lymphs live only for about a couple weeks. They didn't find any significant difference in the life expectancy of the blood cells when it was just the Epstein-Barr virus. But what they did find is when there was a co-infection, when there was pathogen or organism present, that there was a great increase in the life expectancy of that blood cell. So what they did is they took the suspension with its Epstein-Barr virus, some lymphs, which blood cells, white blood cells, which were the lymphs, and this bacteria, Mycoplasma fermentans. So there were three things in the mix there. And what they found is this bacteria, in addition to the Epstein-Barr virus, the two of those microorganisms or microbes cause the white blood cells to live longer. They don't know the mechanisms, but it's interesting to note that they actually added longevity, or the white blood cells became quote unquote immortal. They did not die. Normally they die within 2 weeks. The blood cells did not die when you had both Epstein-Barr virus and the Mycoplasma fermentans bacteria in the suspension. So now this can be good or bad. What does it mean when a cell becomes immortal? Of course, it means that the blood cell doesn't die, but also cancer cells are immortal. They multiply, they do not die. It is basically reproduction gone wild. So they are linking maybe co-infections of the Epstein virus and some strains of this bacteria, which seems to make blood cells immortal. They are linking this to maybe cancer, malignant transformation. So some interesting facts, interesting research topics on some of these pathogens. Anyhow, this last slide here is going to be on the bacteria that causes Lyme disease, Borrelia burgdorferi. So usually these pathogens are extracellular. So once they are introduced into the host, they live outside of the host cells. And they initially will generate some type of immune response. But the response isn't very sufficient. It doesn't kill these bacteria. Instead, these bacteria then have really outsmarted the immune system. They don't even hide so much within a cell, but they will travel to the lymph nodes and set up shop there. They end up infecting the germinal centers of these lymph nodes where the antibodies are made. And the antibodies are made, but they are made ineffective, or they are ineffective against this bacteria. So, in a way, perhaps Borrelia has even outsmarted the Epstein-Barr virus. So anyhow, there is still another implication for people infected with both. Now we're talking Epstein-Barr virus and Borrelia. It seems to have suppressed the immune response to the point that it may be even like comparing it to HIV, where in the case with HIV, the virus interferes with these helper T cells, differentiating, which leads to chronic illness. So we've got the Epstein-Barr and the Borrelia in co-infections. If we've got both in the co-infections, they may even get to the point where they are suppressing all of these type of lymph cells, which will keep them from operating or functioning, and this can lead to many chronic illnesses. So this is just some news and research on not only stealth pathogens, but also on co-infections, being infected with more than one pathogen. On that note, I'm going to end. And I guess I just want to say it's been a pleasure having you. Um, I think if you remember the first day you walked in, many of you did not realize that bacteria did much more than cause disease. But if you look back on the course, I hope you've learned a lot, learned as much as you possibly can given these circumstances. But just realize that microbiology, even though it's a study of microbes, of things that are very, very small, they do have a lot going on. Okay? I want to thank you and hope you all have a wonderful summer.